So that abstract is an oral presentation by one of my associates. We did a study to understand if we could identify any baseline features in patients before the cells were actually manufactured for CAR T-cells. We analyzed the serum proteomics by O-link, 92 proteins we examined, and we also looked at the T-cell phenotype with a number of panels that we developed in-house. And we discovered that there's a signature identifiable in pre-manufacturing T-cells that predict or actually predict response to CD19 targeting 4-1BB co-signaling CAR T-cell. And those samples were taken from a decentralized manufacturing pipeline. And we validated this set also in a Kymriah type product. So now we can predict with fairly high certainty if a patient would actually respond to a 4-1BB co-signaling CAR. And so the signature entails early memory phenotype with CD127, 27, but also we found that there's a type 2 signature like we published last year in Nature in ALL. So this is a shared theme among CD19 targeting 4-1BB co-signaling CAR T cells.